So I better get started. All right, it's 1 o'clock. So it's time for another exciting afternoon of organic chemistry with your host, me, Dr. White, Chem 170. Now, next part of the video, I'm going to cut out because there's stuff I just don't want on the internet for the rest. Put something down if you didn't do that. And also, I mentioned earlier, Board of Trustees. Let's take a little longer than a 5-minute break. Come back at 150, and then we'll talk about real quick a few things. We'll do the lab, and I'll let you go, and I'll send out the password email. I'll see you in about 5 minutes. Time to get started. That one worked better. All right. And before I go any further, I'd like to thank each and every one of you for being in my class this semester. I haven't gotten to know you all the way I do normally face to face, but I hope you enjoy that my world of organic chemistry, which I talked about the first day, is now your world of organic chemistry, and you know about it. And think about it. You know more about organic chemistry than most people on this planet, and use that knowledge the rest of your life. It's one thing my father taught me. Nobody can ever take away education and knowledge from you. All right, just want to go through this again real quick. Everybody see chapter 7 on the screen? Thank you. All right, what's an amino acid? It's a compound that has both an amino group and a carboxylic acid. And a better way to draw it is this. And the amino group, the nitrogen, is always on the carbon attached to the carbonyl carbon, the alpha carbon. Now, when R is not hydrogen, anything else, then on this carbon right here, the alpha carbon, you have 4 different groups, R, amino, hydrogen, and this right here, carboxylic acid. So when R is not hydrogen, the amino acid is chiral. And you put amino acids together to make peptides and peptides to make proteins. And here I say it's chiral. And glycine is the one when it's hydrogen. You don't have to know any names of amino acids. And the Zwitter ion, I'm not going to ask. And here, remember, you take a carboxylic acid, which is this part, and an amine, which is this part, where our double prime is hydrogen, and this part right here, including this R group, is the R prime. You form an amide, which is this. And you learned this for test number 3. So how does Mother Nature put it together, amino acids? By forming an amide bond right there. That's in red. However, protein chemists call that a peptide bond. I'm not a protein chemist, I'm a synthetic organic chemist, and I call it an amide bond. But I'm not going to ask you that. And a peptide, which I won't ever ask, but for other classes you may need to know, have 2 or more amino acids joined together, and the amide bond is called a peptide bond. Now, protein is when you have 50 or more amino acids connect together. Peptide bonds, really amide bonds. And this you should know for the final. This won't be on test number 4. What's the bond between amino acids and a protein? An amide. And you learn if you take an amide and react it with HCl and water, why HCl? Excuse me, that's the acid in your stomach. And you get back the carboxylic acid you would have used to make that amide. Plus, since amines are basic, you get the amine salt. of the amine you would have used to make that amide. And if you had, here I just have 2, but if you had 50 of them, same thing happens. Here's my amide. This part was the carboxylic acid. Now this amine is a base. The presence of acid will form the amine salt. You learned that for test number 2. However, this amino group is this one right here. And essentially you get back the amine salt of amino acids, which makes them more water soluble. You know, like dissolves like. And what this really means is, and I don't know if I think I mentioned it, and I'll do it again now. You should know now for the final, and also leaving my class, how to describe with words in a general chemical reaction what happens when you eat a protein or something containing protein, like chicken, or if you're a vegan, soybeans. And how do you describe it with words? Well, what functional group is key in a protein? And that's amide. And in your stomach is hydrochloric acid and water. So you have acid hydrolysis of what functional group? And amide. And the general reaction, I'll put it down, but you don't have to. Our prime and our double prime can be hydrogen. The general reaction, as you learned for test number 3, is an amine salt plus the of the amine you would have used to make that amide, plus the carboxylic acid you would have used to make that amide. And with that, I'll be doing the lab in a second, but this is important. First of all, I've done everything this semester in terms of teaching you material. We're done. However, next week, I'll be doing the whole week, reviewing the whole semester. It helps my students show up. It will help you. Now, a couple things over the weekend or by Monday, hopefully, one, I'll get the test graded like I did the other test and send out your in Blackboard announcement, hopefully by Sunday noon, if not earlier, your test score. And by Sunday afternoon, I'll send out the individual points. On Monday, I'll go through the test number 4 answers, but I will cut that out of the video. How do you like that? Is that cool or never mind? But anyways, I'll send out the answers. Now, I'll also post the breakdown of the final exam. The final exam will cover the whole semester. It'll be 210 points because there's going to be 10, yes, 10 bonus points. Also bonus points. Don't forget, if you're doing the extra credit project, get it in today sometime. Upload it to D2L. All right, now, how do you study for the final? You all have my exams. Use those as practice problems. Am I going to put the same questions on a final? No. But am I going to put similar ones? Of course. Look at my practice problems. Aren't they similar to what I put on a test? Yes. So that's the best way. And also study notes so you can look at me on the video too. See, I'm a video star. All right. About the final exam, I'll do the test point breakdown for tests 1, 2, 3, and 4, and how many are nomenclature, how many reactions, general knowledge. Now, one of the things I do, and don't tell anybody I'm being nice, but the first part of the final is the material from test number 1, then test number 2 material, test number 3, test number 4, and the stuff on amino acids and proteins, which will be a couple of points. And that's the final. Now, if we were face to face, you'd have 2 hours to take it. Most students are done in an hour and 15 minutes, assuming you practice and study. I've had students who have, and they can take all day and they're not going to finish because they don't know what's going on. It's only happened a few times, thank you. But anyways, that's that. Any questions before I move on to the lab? Looking down at chat, nothing popped up. Listening to my speakers. All right, it's time for the last lab. And this last lab deals with carbohydrates, but more specifically, starches. And one of the things I've done all semester in the lab, and if we were in face-to-face, you actually see it when you were doing the labs. That's the sad part, but I had a decision to make. Is it worth doing half the labs and risk your health going to ECC and mine too? Or do what I've done, which have all virtual and not do half, but do all the labs, and you get to learn things. By the way, Gesundheit, Justine. In a class, I would have said that too, and then said my grandmother, that was Grandmother Greenman, what a great woman, taught me well. By the way, she had a dangerous sense of humor. For those who want a Grandma Greenman story, wait after class today, and I'll be done in about 10 minutes, 15 minutes. I'll tell you the Grandma Greenman story. What a great lady she was. But anyways, I was lucky enough to get to know her till I was about a teenager. But anyways, chemists until the modern electronic and analytical equipment back in the late 1800s through 1950 would develop visual tests to help identify things. So how do you identify? And think about it, late 1800s, you're learning about food and everything. Does this have starch in it or not? And that's important. It's a carbohydrate. Well, they came up with a color test. Everybody see my whiteboard? Thank you. Now, a carbohydrate, a starch, is a polymer of glucose. Now, in real life, it's not a straight line. It's a helix, like this. And along here, you have your hydroxyl groups from the glucose. Remember, glucose, which I didn't ask you to learn the structure. And the cyclic form. Oops. I just did the fructose one. I need to do the glucose one. And this is connected to other glucose. So you got these hydroxyl groups there, right here, which are all along here. Throughout that whole, the spiral. Now, someone found out if you took iodine, which is a red-brown color, and you put it inside here, this now turns to dark blue. So dark, it looks actually, unless you look at it under good light, it almost looks black. However, this is a solid, and that's hard to deal with. So someone found out the following, and that is if you take iodine and react it with potassium iodide, you get this potassium, and here you'll learn my K plus I3 minus potassium iodide, whatever. And this is now ionic and water soluble. And now you can make a solution. Actually, they also put some ethanol in there to help make it more water soluble. And now the same thing happens if when I'm in a starch, oh look, curly French fry, never mind. And you have the iodide anion in here. This is a reddish brown. I think iodine is more purple. I take that back. But this is a reddish brown, almost more brown. And inside, this also turns dark blue. And it looks almost black. It's that dark blue. And if something turns potassium iodine solution, which is also called tincture of iodine, and one of the reasons they mix it with ethanol, that water in the Ki3 minus is very good at killing germs. And when I was a kid, everybody's mother had tincture of iodine in the medicine cabinet that if you were scraped your knee or your elbow or your hand while you're outside playing rough with your friends, or if you cut yourself, come here, I got to put this on. And because of the ethanol in there, it sting like all heck. And later on, somebody invented another biocide that was in water alone. They called it Bactine, and they sold it because they sold it to mothers on TV. It doesn't sting like tincture of iodine. Anyways, we have tincture of iodine. And what you're going to do this lab is find out if things are starch or not. And what things are you going to use? These. And what we're going to do is take tincture of iodine, which Ki plus I2, which makes this now water soluble, will turn blue or dark blue from reddish brown color when added to a starch. And this is due to a complex iodine ion trapped in the helix form. And in the lab, by the way, COD does, it's the wrong school, ECC, some of these things they don't buy, so I bring it in, or I have other chemists who have it, bring it in. And you'll notice if you put it on a potato or bread, it goes dark blue. Quick Dr. White story, oh, I got time. Because I'd bring this in and I wanted to leave bread in the lab and only do it once a year or so, bread itself is going to go moldy. What's something that doesn't go moldy for a long time? Matzah. And being Jewish, yes, I always have matzah around, especially when it's cheap to buy in Passover, I stock up. So I would crush up matzah. It does the same thing as bread. And funny story, the first time we did this, Dr. Daly, who's the lab manager, who also teaches organic chemistry the 2 semester, who's a tremendous job. And he's also very meticulous, which is a good thing for us faculty and you, the students, labels everything. Well, the first time we taught it, he put on the bottle that had the broken up matzah. And the students were looking around and said, Dr. White, where's the bread? I realized, why? I've got people who've never seen matzah before. And after that, Dr. Daly changed the label, bread and a bracket underneath matzah. Now, I would have to bring in the cheese. And the first couple times I brought in some decent, not cheap cheese. Then I figured, why am I spending all this money on good cheese for this lab? I can just get what's ever on sale. So I got the Kraft-like cheese that's individually wrapped. And I happened to bring the label in to show the students in case I hadn't looked at it. And students said, Dr. White, the yellow cheese is turning dark blue. How can that? There's no starch in cheese. And I said, all right, let me take a look. You run it. And I watched it, and sure enough, turned dark blue. I picked up the label from the package, and it had starch filler in there. That's why it was cheap. And our test showed that. And if you get a better quality white cheese, named after me, not really, you'll see it doesn't have any starch in it. And notice we do flour, cereal. I think Dr. Daly brought that in because I'm not a cereal. I think it was Cheerios, and sugar, honey, and cornstarch. And here's the results. And here are the questions that you have to do. And with that, I'm done, except what was the story I was going to tell? I just lost my train of thought. Somebody help me out. What was the thought I was going to talk about at the end of the class? Come on, somebody tell me. You remember, don't you?
Your grandma?
Ah, thank you. I forgot. All right. Grandma Greenman was a dangerous lady, had a sense of humor that was dangerous. And plus,
I got to interrupt you for a second.
what?
My wife just shouted his grandma story.
Tell your wife, thank you for the assist. Because it just went out this year and went out that way. Anyway,
She's paying more attention than I am.
well,
No.
she can't help you with test number 4, though.
Okay.
Anyways, Grandma Greenman was one of the finest cooks I've ever met in my life. And back in for holidays, she'd have my family over, my mother, obviously her daughter, and I had 2 uncles who and their family would all go over to Grandma Greenman's for holidays, Jewish holidays, and Passover. Now, when I was about 5 or 6, I don't know when it started, the night before, my parents would drive me over and I'd spend the night with my grandfather and grandmother. And they were great people. It was fun. And Grandpa Greenman was dangerous too. Oh, you smoke cigars? Yeah, here, try it. And I never smoked a cigar again. And then, oh, what are you drinking? Oh, Canadian Club. Here, try it. I like that. But anyways, she was cooking. And one of the things for Passover you need is horseradish. Now you can go to any store in any supermarket and go to the dairy area. Usually they keep it near there and buy jars of horseradish. Well, back then you made your own. So you get the big horseradish root. And one time I actually did that later on my own. And it's pretty intensive. Horseradish root is strong. And back then she had a hand grinder and she peeled it. I'm watching and she's grinding it, collecting, putting it in a container to use later on for it's called the Seder. I said, Grandma, what's that? And she said, horseradish. And said, oh, what's it like? And she had that look on her face I hadn't learned. Be careful. It's Grandma Greenman. I'm going to do something to you, time. And she said, here, smell it. This is pure horseradish, freshly ground. I go like this. Next thing I knew, I was on the floor. And she's standing there laughing and said, teach you to stick your nose into something you don't know what it is. And she was laughing. Yeah, but she made her own gefilte fish, and that's my Grandma Greenman story. There are others I could tell you, but that's a good one to end the day on. With that, I'm going to say, gang gesund. Bye now.